Here we have one example of a BLAST result. At the top you have information about the query sequence that was submitted to the BLAST server. Move down and the graphic summary of the results. At the top the information is for the query sequence, and as we see here, the length of the sequence that was submitted to BLAST was a little bit more than 1200 nucleotides. Below is a series of alignments, basically sequences from the nucleotide database that were identified as being similar to the query sequence. The information is organized in such a way that the better quality alignments are provided first. If I left click on the very first alignment, I see the score 2274. If I go below, I left clicked and the score is now below 2000. It's 1916. So the higher alignment scores are provided first, and you also have the information is also color coded, which means that all alignments with all alignments that have a score above 200 will be displayed in red, and you also have information for the other colors. Here is if we have a thin gray line with 4, 1, 2, 3, 4 alignment boxes, and this means that the subject sequence that was identified within the BLAST database had 4 regions that could be aligned against my query sequence, and the thin line is for a gap. So in this case there is a gap, so the nucleotides are missing from the subject sequence that was aligned, but they are found in the initial query sequence. Now if we move further down, okay, we get alignment results again. There are options here that are not available unless you select some or all of the alignments, and once you've selected some of the alignments you get access to other options. And for each alignment it gives you the alignment score, it gives you the query cover, so how much of the alignment range covers the initial query sequence. In this case it's a full alignment in the way that the alignment box covers the full length of the query sequence that was submitted. The E, the expect value, which is a sort of a statistical analysis, and more information as described in the worksheet. And then for each of these alignments, if you move down, you'll get the specific sequence details. So you get the nucleotides and you see whether you have a match or a mismatch at the different positions within an alignment box. So this is an overview of the BLAST result. Make sure that you know how to interpret the score value, the query coverage, and the P or the expect value. There'll be many questions on the post-lab quiz that will refer to these parameters.